Hello, everyone. Glad you can join us. Feel free to say hi in the chat, especially if you're new. Let us know where you're coming to us from. We're going to do a few Rosalind problems today. We actually have 7 Rosalind problems today, and we're getting towards the later stages of the bioinformatics track on Rosalind. So as a result, it means we need to be a little judicious about time and moving through these quickly. So I'll just remind you that our model for these is usually I code an entire problem, and if you have any questions, feel free to let us know in the chat, and I'll address those in between questions. We're eventually going to cut these down, I should say, into individual problems. So all of our videos on demand are available on our YouTube channel, so you can check those out. Please make sure to subscribe if you haven't already. And then we'll be putting out shorter capsule problems so that you'll be able to find an individual question if you're interested in that. I'll also be putting out some messaging around our schedule for these streams going forward. I think we have like 4 streams to go, maybe 4 1/3. There are a couple problems that are remaining from what we've done before that don't really fit with the themes of some of our streams, so I might just put those out as bonus content. But we have 4 to go, and I have our high school program. We have a pre-college program in computational biology that starts on Monday, and that's a pretty hectic time for the next 4 weeks, where we bring in 100 students from around the world, around the country and around the world, to learn all about computational biology and the intersection of science, AI, and experimentation, which is a fun time. So that will be, look out for a message from me. If you're not on our Philomath mailing list, please make sure that you join that. And there's a pinned comment here, and there's information in the description around how to join our mailing list, how to join our Discord, and how to join our Philomath organization as a member. So we have a member-only course starting on Monday too, our Vibe Coding Studio, where you can bring a project that you've been really interested in, and we're going to build a community of learners. I'm going to explain kind of how I think about learning and building in an era of AI, what AI even is. We're going to talk about all that over a 4-week period, and then you can kind of build your own project for a period of 6 weeks over the summer. So that kicks off on Monday. The subject of today, of course, is solving 7 questions on Rosalind all around population genetics. So we're going to get started very shortly. There's starter code in the chat. There's completed code in the chat, or not in the chat, in the description of the video. So you can find all that as well as links to everything else that we're trying to make, all in the description. Please make sure to like and subscribe if you haven't done so already, and drop us a comment, keep us visible, all that good stuff. Again, if you have any questions as we go, let me know. I'm always happy to handle those synchronously too. But we're going to start with INDC. That's going to be our first problem. And I'll get started here shortly. So without further ado, let's code. All right. INDC is independent segregation of chromosomes, also pronounced indica. The idea behind this problem is Mendelian genetics. So most likely, you and I share something, which is that we have 23 pairs of chromosomes. And in each pair, you're going to receive 1 from each of your biological parents. And they receive 1 from each of their biological parents too. So each of your parents has 46 chromosomes. You get 23 from each parent to combine into your 46. And critically, it's not just any random collection of 23 chromosomes. You get 1 for each parent, you get 1 from each of their pairs. So there is a process when gametes are formed that you can think about as almost like flipping a coin. heads or tails, where one of these two chromosomes is selected when that gamete is being formed. And so I want you to imagine for a moment that you could have a sibling who has a collection of chromosomes too, and they get 23 out of the 46 from each parent as well, but they're not necessarily the same. There is a small chance that you actually got, from your biological mother and your biological father, you picked 23 in each case, and your sibling picked 23 in each case, so 46 total. Over all 46 of those coin flips, it is at least conceivably possible. We know what the probability would be. It would be 1 over 2 to the 46, which is a pretty small number, but it is conceivable that you could actually have obtained the same choice of chromosomes as the sibling. And in that case, in theory, you'd be an identical twin. Now, things like recombination are going to prevent you being an identical twin. However, the idea here is siblings share about half of their DNA on average, but in terms of chromosomes, there truly is a random process that's going on. So you could have more or fewer chromosomes in common with a sibling. What this question is going to ask is, well, can we study that as a probabilistic process? So what's the likelihood if we think about this as flipping a coin some number of times? In this case, say you flip it 92 times, what's the likelihood that you would obtain 51 heads versus 27 heads, or at least 33 heads, and so forth? And that brings us to our problem, which is going to look at a binomial random variable, which models this idea of having n coin flips, perhaps where there is a weight to the coin. So we're going to look at an initial problem here where we have a probability of 1/2 as the coin coming up heads or tails. But you could imagine that maybe it's 30% that it comes up as heads, and we're going to generalize this in a later problem. But for now, we're going to say that this random variable has an underlying probability equal to 1/2. And our input for the problem is going to be some number n. That's going to be the number of coin flips. And the output is going to be an array that represents essentially the probability of having that number of coin flips come up heads. We're going to take the logarithm of this probability because some of these numbers are going to be quite small. And that will allow us to not just represent these numbers as all 0, but the array is going to be of length 2ny. Well, we're going to take n. The idea is we're representing this kind of as a biological question. You're a diploid organism, meaning you have 2 copies of each chromosome. So you're going to have 2n total chromosomes. And what A of k represents is this logarithm of the probability that if you take 2 siblings and you view this process of just choosing, for each parent, choosing 1 chromosome from each pair, what's the likelihood that 2 siblings are going to share at least k of the 2n chromosomes? So as a result, if we think about this being 5, notice that you have an array here, and this array has length 10. So this is the likelihood of sharing at least 1 chromosome, 2, 3, 4, 5, 6, 7, 8, 9, and 10. I guess we could have an extra value that's sharing at least 0 of their 2n chromosomes, but that's always going to have probability 1, which means the logarithm will be 0. So this problem doesn't ask for that value. That's always just going to be 0 at the start. As we said, our idea here is that each choice of chromosome is essentially this 50-50 coin flip. Did you, for all of your 46 or 2n chromosomes, did you have the same choice of chromosome as the sibling? It's the exact same question as saying, if you flip a coin 2n times, what's the likelihood that it comes up heads at least k times, if we're saying you're sharing at least k chromosomes? Now, if we were instead to ask, what's the likelihood of sharing exactly k of these 2n chromosomes, then we've already seen this question. We introduced this much earlier in the question LIA. And in LIA, what we saw is that if the coin has probability p, this was a general problem that we looked at. If a coin has probability P, then the likelihood of exactly, in this case, I heads coming up when flipping the coin is equal to this expression. So it's P to the I times Q, which is 1 minus P, times N minus I. And then we have times the combination statistic of N, here, which is represented as population, choose I. So this is the binomial distribution, and this way of determining this probability is based off of this binomial coefficient of N choose I. And our solution here is going to be based off of this. So what we're asking for here is really to generalize this idea, to compute this probability of at least K coin flips coming up heads, and to put this into an array where we're computing all of those. So this is very much a warm-up question for our other questions in population genetics. And our function that we want to write is called share at least logs. It takes an integer n as input, and it's going to return a list of floats. Those floats are going to be the probabilities that we care about, except we're going to take the logarithm base 10 first, so that any values that are close to 0 are instead just going to get converted into negative numbers. It's a common thing to do, so we avoid everything just seeming like it winds up being equal to 0. So the first thing I'll do is write down, remember that when we take in an integer n here, we also need to write down the total number of trials that we're going to have. And that's the chromosome total in this case, which is just 2 times n. So you have a total number of flips, in other words. Then it's just a matter of saying, well, we're going to first find all of the probabilities. So first, we're going to look at, if you do 2n coin flips, all with probability 0.5, what's the likelihood you're going to have exactly K heads? So we're going to build an array that stores all of this. I'm going to call this point probabilities. It's a list of floats, and it is going to be equal to an empty list initially. And then I'm going to go through and set these values. So for I in the range over chromosome total plus 1. So we're going to range between 0 and chromosome total. And I'm going to say, well, the point probabilities that we care about, we're going to append. Here's where we get to call our math combination statistic. So it's going to say chromosome total, choose I. And then we want to multiply this times P to the I, which is 0.5 to the I. And then we want to multiply it by 1 minus P raised to the N minus I. So in this case, it's chromosome total raised to the 0.5 times chromosome total 0.5 raised to the chromosome total minus I. There we go. So this is doing quite a lot, but really it's saying, you know, the current probability is equal to, let me expand this out so we get a little bit more. We're setting this equal to this just for readability. And then we're going to append that current prob. The other thing I want to point out is you might notice you have a 0.5 to the I and you have a 0.5 to the N minus I, and those things are multiplied together. So you have a number raised to a power multiplied by that same number raised to another power. So we can add the exponents. When you add I and you add to chromosome total minus I, well, the I's are going to cancel. So as a result, we get the same thing by deleting this. This is only because P and 1 minus P are the same. And now this is just going to be chromosome total here. And that's our formula. And now this gives us the exact probabilities of equaling k heads in 2 times n coin flips. Now what we do is, for each value of k that we care about, sum all the probabilities that x is greater than or equal to k. And take the logs. So we're going to do both of those steps in one. It's just not a lot cleaner to have the probabilities that we care about first. So I'll say the logarithms are going to be a list of floats. We can start off those as empty. And then we'll say for k in range. Here I'm going to range up to chromosome total plus 1 again, because we have n plus 1 possible outcomes ranging between 0 and n. Except that remember that the problem itself doesn't care about the likelihood that x is at least equal to 0, which is just 1. So we're going to start our ranging here. And then after we do this, we'll say, all right, let's set the tail probability equal to 0.0. So that's where we'll start with. So this is going to be our eventually, what we'll do is logs.append math.log10 of this tail probability. And then the idea is, we're going to range over all values greater than or equal to k and add probability into the tail prob. So for I in range, now the range is going to start at k and still go up to the chromosome total plus 1. As we do that, we're going to tail prob plus equal point probabilities of I. And then at the very end of this process, I'm going to return logs. So we compute the entire probability first. Might be nice to do the log as we go, but the issue here is we want the logarithm of the overall probability, and that probability is computed as we go. If we were taking the logs as we go, we'd be doing log of a sum. Log of a product is nice because log of x times y is log of x plus y, so you can just sum logs as you go. But if you're doing the logarithm of something as you go with a sum, that's not going to work. So as a result, we need to compute our probability that we care about first. And then at the end of this, take the logarithm of that resulting probability. So hopefully our problem is solved. Got an expected value here. And I can run Python 3 indica.py. Okay, I have a mess here. Let's see. Try that again, a little extra parentheses. And everything looks to be reasonable here. Okay, we have a classic thing of positive 0.0 versus a negative 0.0. So that's a common programming thing that you'll see of this number being close to 1. So when we take the log 0 and negative 0, these numbers, or when there's a round off, numbers can be very close to each other. So a lot of times you'll see the sign incorrect, but everything should be okay. I think the platform will handle that. So we're ready to download the data set. And once we've got our larger data set, I'm going to put it into our folder, call it dataset.txt. Make sure I get rid of our old data set here. And I forgot to uncomment out this, so let's do that. Good. And even though we had a lot of negative zeros there at the start, everything was okay. I was holding in a cough for like 10 minutes there. All right. Any questions on that, let me know. Josh, welcome. Yeah, if you have a quick question about the vibe coding course, you can. Probably a lot easier to just join us on the Discord and ask questions there. But we're going to look at AFRQ next. So get set up in the meantime. Yeah, Josh, good question. And I'm going to put out a message around this. We've been busy times, but there will be messages for our members kind of around what to expect. We put out an initial message. Everything here will be on YouTube Live. We're going to meet once a week. Actually, the first live will be open to everybody, and then the other 3 are going to be subscriber only. So if you want to try it out, you're welcome to, if you haven't joined already. But yeah, what I'm going to show you is the idea is not to try and teach a tool, right? Like, and I would say there's a good analogy for this, which is I've never been wild about caring what programming language something is taught in. So when we did Programming for Lovers, our stream series earlier on in the year, I did Python because that's what people wanted. I don't think it's the most fun necessarily to teach programming from. I mean, these streams are fine. Python's great for this, I think. But for teaching fundamental programming, I don't think Python is a good choice of language. And at the same time, I don't also really mind. So, I think that there are things to learn about how to program effectively, regardless of what language you're in. And there are hallmarks of things that go back many years. So that's a lengthy answer. But yeah, I will show a certain stack that I use, which is going to be, something like Notebook LM for doing research on a problem you care about, how to set up a Claude environment so that it functions as a second brain that you can offload things to, and manage things effectively. But I don't really mind what particular tool that you bring to it. It's just sort of like, I'm going to show you what has worked for me. But that's not meaning that I am teaching Claude, because things are changing so fast, although I will use it as a model for teaching, I hope, evergreen higher level things. So bring what you like. That's a great question. I'll make sure that that's something that we talk about. But the first lecture that we do on Monday, I mean, we're not jumping straight into AI tools and setting up a stack. I'm going to talk about what is a neural network and what is an LLM and why do we care about this and why does it produce output that's sometimes like incorrect, et cetera, et cetera. So that's where we're going to start, and then we'll go from there with a lot of the more practical side of things. So I hope you'll join us. In the meantime, we're going to move on to our next problem. AFRQ is counting disease carriers. We've seen Mendel's laws. Mendel's laws are cool and all, but they don't get to population level changes. So we would love to study on a higher level, not just Punnett squares, right? But how do we appreciate how alleles travel throughout a population? And there is a lot of foundations of the study of population genetics in the early 20th century. Hardy and Weinberg laid out kind of, you're at genetic equilibrium when there are changes in the population, of course. But for one, the population is so big that the random changes in the allele frequency, which we will study in other problems, are essentially negligible. So if 80% of the population has an A at this particular position of the genome, for example, and 20% have a T, changes in that frequency of alleles is not going to vary that much from one generation to the next because, well, the population is huge. So in humans, for example, I think that's probably a reasonable assumption because we have several billion members. Now, that's possibly not a strong assumption over a very long time scale. We'll talk about that in a different problem. But we're also assuming that the gene's not really undergoing a lot of mutations. And similarly, it's not being selected upon. I think those two concepts go hand in hand. So it's not inherently important to survival, for example. And then gene flow is negligible too. So that's due to migration into and out of the population, change in allele frequencies because of that. That's not happening either. And then mating is functionally random with respect to the gene that you're studying. In all populations, sometimes that's a good assumption, sometimes it's not. So that there's a lot to be studied around this set of assumptions and when they're not true. But the idea is let's start with a simple model. And this is a hallmark of computational biology and data science in general, which is to understand something, let's make some simplifying assumptions, and let's understand it in those simple cases first. Then we can always relax each of those assumptions and see how they change things. So Hardy-Weinberg is saying, if you're at genetic equilibrium, then the frequency of the allele for which you're studying is going to remain constant and essentially evenly distributed throughout the population. So we're no longer studying individuals. All we're really studying is the frequency P of that allele. And by saying that, I think the idea is we're not far away from this same concept that we saw in previous problems, which is, oh, well, if the frequency is P, or let's say 80% for our previous example, then I suppose a new individual coming from that population has a likelihood then of 80% or 0.8 of receiving that allele. So there's again a weighted coin flip behind the process, behind the scenes. And so this model is trying to move us towards a binomial random variable again. We want to model this principle. And so we want to assume then, again, we have a collection of diploid individuals, and they have 2N chromosomes. It's actually a typo here. It's not N diploid individuals. It's a collection of individuals having 2N total chromosomes. Oh, excuse me, never mind. I thought I had a typo. No, it's individuals, which means there's 2N total chromosomes. So if there are M total, either dominant or recessive alleles, it doesn't matter. Say that there's M dominant alleles. So before our example was like we were trying to get to 80%. And so what that was saying is if 80% of the total chromosomes in this population have this particular allele. So if you're selecting a chromosome, you're drawing one out of the hat, or you're flipping a coin, say what's the likelihood it's going to be a dominant allele? It's the total number of dominant alleles that you see divided by the total number of chromosomes, M over 2N. So that P is equal to M over 2N. Remember, we're assuming that the population is huge. So it's kind of like we're assuming what happens as N goes to infinity. So right now, we're only going to look at this value P. We're not even going to think about this particular study as a binomial process because N is so huge that it's kind of heading off to infinity, and all we care about is the proportion. So what we're going to be given is this array, A, that represents the proportion of individuals for each of a collection of factors that are homozygous recessive. What does that mean? It means that they could possess 2 copies of the recessive allele for this factor. And we're assuming that we're in genetic equilibrium. So you're really just getting a bunch of different values of P. You're getting whatever this P computation is for many different genes or many different factors, however you want to frame it. And what we want is an array of the same length where the kth element of this array is the likelihood that a randomly selected individual from this population is going to carry at least 1 copy of the recessive allele for the kth factor. So if there is a practical side of this problem, it's that, well, if this is a genetic disease, for example, that's a recessive illness, so you only actually express the illness if you're homozygous recessive, you have 2 copies of this, you maybe can measure the number of individuals in the population who have that condition, have the disease. And then the question is, for a collection of different conditions, could you estimate the likelihood of someone being a carrier? So you're going to, for these recessive conditions, you're going to have way more people who are carriers than who have the illness. And the problem is asking for the probability that somebody has at least 1 copy. So they could have 2. You're going to have some collection of individuals with the disease, but you're going to have a lot more people who have at least 1 copy of the allele. So what this is telling you here is, for the sample data set, is actually something that is, I think, from a population study, quite powerful. It's saying if you have a recessive illness and 10% of the population has it, 53.2% of the population are going to be carriers or have the illness. So that means 43% of the population are carriers, meaning they only have 1 copy, so they don't have the disease, but they are carriers. At 25%, it becomes 75%, and at 50%, it becomes 91.4%. So the idea is we can actually compute these values. And what's nice about it, this particular problem, is that because we're not even thinking about coin flips, and I'm just thinking about the value of P that's being treated as stable, these carrier probabilities actually just boil down to some pretty simple math. So the idea then is, at equilibrium, we're going to say that the probability that, a randomly chosen person has the disease is equal to P squared. So however we write it, P caret 2 or P times P or P star star 2. And here is where P is, the proportion of the recessive allele in the population. So the idea is every person is formed from 2 random people, and we're assuming that mating is random. And so you're essentially just, there you have so many possibilities. You're picking 1 chromosome here, 1 chromosome here. They each have likelihood P of having the recessive allele, and you have the disease if you get both. And as a result, it's just a product rule. So it's P squared. So once we do that, what this means is, 1 minus P is the proportion of the dominant allele in the population. And those are good things to write down because we want to combine these. So we can get the value of P simply by taking the square root of both sides, because if we know the number of people in the population with the disease, we know the total number of people in the population, we already know this value. We know the likelihood that somebody has the disease because it's just a ratio. So what we want is P first. So P is equal to the square root then of this probability, which we can observe as a frequency. I just took the square root of both sides of this equation. And once I know that, well, 1 minus P is going to be this proportion of the dominant allele. I'm just copying that down again. And so we can easily compute each of those 2 things. And remember, well, we want to know what's the likelihood of somebody having 2 or 1 copy of the recessive allele. And it is far easier to say, what's the likelihood of someone having no copies of the recessive allele. And so this would be 2 dominant copies. And in that particular case, well, it's the likelihood of getting 2 dominant alleles multiplied together. So in that case, it would be 1 minus P squared. And that's helpful because the probability then of at least 1 recessive allele, these events are complementary. We've seen this previously on the platform, right? You could have 2 events, and if they're disjoint from each other, and they together constitute the entire probabilistic space of possible events, then they have to sum to 1, so that we know that the probability of at least 1 recessive allele, plus this value that we just computed is the probability of 0 recessive alleles. Those two events are going to constitute, they're disjoint, they constitute the entire set. So the sum of these two probabilities is equal to 1. We wouldn't write it that way in practice, because what we would just do is we would move this to the other side of the equation, and we would get that the probability that we care about is 1 minus this expression that we computed before. So it's just a matter of ranging over the dictionary or the list of floats that we have, which represent this, and then computing this value. So you take the square root of what you see to give me a value of p, and then for each factor, we just compute this value. So again, powerful stuff, and I think a great initial indication of how we can use a little bit of math to take what we observe about a population and what we know biologically and be able to infer something that might not at first glance be easy to infer. And we can do this for as many factors as we like. So we're going to say results is a list of floats, and it's empty. And eventually that's what I'm going to return. Return results. And in the meantime, I'll say, all right, great. So for each frequency value n, we're taking as input these recessive phenotype frequencies. So as I range over that, and here's where we will say, we know what we care about. The recessive allele frequency, remember, is equal to, you have to have 2 copies of the recessive allele in order to demonstrate the disease. And so we have the probability of the disease, which was what we're given. This is going to be our p. And in fact, I could write this out as recessive allele frequency, and then I'm going to have dominant allele frequency and so forth. I don't know. Because of how I've written it out here, I'm actually thinking maybe I just write it as p. P is what we really kind of care about. And p is just math.square root of the frequency. And I can comment, this is the recessive allele frequency. And I could be very precise and say the dominant allele frequency is 1 minus p. And then, the likelihood of, being homozygous dominant is that 1 minus p times 2, or raised to the power of 2. And then I subtract that from 1. But if I understand, if I appreciate all that, really what all I want to do is to say results.append, okay, 1 minus, and then this expression here. So we just say 1 minus p squared. And hopefully that should work for us. So this winds up being a pretty short problem. And we'll see Python 3, AFRQ.py. And that gives us the same values from before. So we see that this is matching on these 3 inputs that we saw. And so once we have that, then we can go to the platform, get a data set from Rosalind. Put it in our folder. Call it data set. And we comment out, or uncomment out the parsing part of our code that we provide. Run it again, and then we're going to get an output. All right, all good. Success. A little bit of math, but a powerful conclusion. Now, there's a lot more to this. Geography can influence things like this. A lot of this stuff is tied to ethnicity. There are always going to be complications, which means there's always going to be areas of interest with studying population genetics. So it's still a very active field, but this was a powerful initial thing to be able to say, let's study make some simplifying assumptions and see under those assumptions we can actually make a powerful conclusion just from what we can observe. All right, moving on. Let us know if you have any questions. We're going to look at the next question, which is SEXL. Hi, Sarah. Welcome. Maka, thanks for joining us. All right. Just do a little cleanup over here. Okay. SEXL is sex-linked inheritance. We've talked about chromosomes. We've talked about pairs of chromosomes. In animals, one pair of your chromosomes, the final pair, are sex chromosomes. Everyone will have, typically, at least one copy of an X chromosome. Females have a second copy. Males have only one copy of an X, but a Y chromosome that's much shorter, and that constitutes essentially the, organisms are female by default, and then the Y chromosome is the maleness sex chromosome. And we've also started talking about this population study of alleles with Hardy-Weinberg. But things get weird a little bit, and the probabilities and the study of things becomes a little bit different when we're looking at alleles on sex chromosomes, because an allele on the Y chromosome, for example, could only be possessed by males. So it's impossible for, if there were a genetic condition affecting the Y chromosome or just an allele on the Y chromosome, it would be impossible for a female, if we're going back to studies of Drosophila or fruit flies, it would be impossible for a female fruit fly to display that allele. Similarly, though, if you're on an X chromosome, things get weird there too, because, well, males would only have one copy, but females would have two. So if you imagine a recessive condition that's on an X chromosome, a male would be more likely to display it because they only have one copy, and the females would have two copies. So they would have two chances to have a dominant allele and display that. We kind of take for granted, I think, the foundations of genetics, and I wouldn't say that it's lost. I think a lot of people have appreciation for it, but I mean, for one, the amazing experiments that actually were able to see chromosomes that go back in some cases the 19th century, and just as simple of a conclusion as what happened in 1905 with Stevens and Wilson finding that one chromosome is shorter, but the female animals had two long chromosomes. Like that, at one point in time, that's a revolutionary idea. Similarly, the real revolutionary idea with genetics, I think, people focus heavily on Mendel. A lot has been said about, by modern standards, Mendel's experiments might not be that statistically kosher. He cherry picked a lot of things. And, a lot of what Mendel concluded is violated for certain types of traits. And I think a lot of the truly foundational work in genetics wasn't necessarily Mendel. It was what was happening mostly centered at Columbia University at the turn of the 20th century. And, one of the titans here is Thomas Hunt Morgan. And that's because he very thoroughly tried to place genetics on a statistical footing by studying fruit flies endlessly. And, here is sort of one of his experiments on fruit flies that just looked at eye color and, was able to establish that this is a sex-linked trait because the probabilities that he obtained don't match what Mendel's work would have predicted. So Mendel's work is full of exceptions. And here's an example of it that shows that this trait has to be on one of the sex chromosomes because you get different results for males versus females. I'll explain this on a very high level because it's a beautiful experiment in biology to show that this has to be a sex-linked trait. So it turns out that this allele for white eyes is on the X chromosome. And it's a recessive allele on the X chromosome. So what that means is a female has to have 2 copies of it in order to display it. A male only needs 1 because the male has a Y chromosome. So as a result, that this male with only 1 copy would be white-eyed. So Morgan crosses a homozygous dominant red-eyed female. So they have 2 copies of the red-eyed allele with a white-eyed male having 1 copy of the recessive allele. And then produces males and females that, the males are certainly getting a dominant copy. The females have 1 dominant copy and 1 recessive copy because they're receiving only an X chromosome from the white-eyed male, but these males receive only the Y chromosome. So there's no way they can be inheriting the recessive allele. Then when you look at the second generation and you cross these organisms again, what you see is, well, you have on the left side, these are your female organisms, they're only red-eyed. And the reason why is, as you can see, well, they may get the recessive allele, but they're not getting it from the male. They're only getting a dominant allele from the male. So there's no way they can display white eyes. But on the male side, that's not true because even though you had a red-eyed male parent, maybe you get a red-eyed allele from the mother, or maybe you get the white one. So even though this was a red-eyed male, it turned half of its progeny would be white-eyed. So you can see from this simple experiment that there must this even if you haven't worked out exactly where this allele is and that is on the X chromosome, you would be able to conclude that it's sex-linked immediately from this because your proportion of traits in your offspring varies depending on whether it's male offspring or female offspring. So in this case, you started with this circumstance and you wound up with 100% female red-eyed offspring and 50% white-eyed male offspring. So you've established conclusively that this must be a sex-linked trait because otherwise, well, you would have equal proportions. And then the second experiment here is you have a white-eyed female, so it has 2 copies, and a red-eyed male, meaning it has 1 copy of the dominant allele. And when you cross them, you get females that are going to have always red eyes because they're receiving this X chromosome from the male previously. And you're going to have males that only have white eyes because they're receiving one of these 2 copies over here. So again, that's strange. That in and of itself would be able to demonstrate that this is sex-linked. And then when you look at the next generation, again, the same type of argument applies. You're going to get 50% white-eyed females on this side, and you're going to get 50% white-eyed males on this side. So it's kind of bizarre that you get the same proportion down here in the second generation, but you get a different proportion up here in the first generation. So notice that these pictures are flipped. In this particular case, you get equal proportions in the first generation, but unequal proportions in the second. Here you get equal proportions in the second generation, female to male, but unequal proportions in the first generation. So if you just did this experiment, you would know that this must be a sex-linked trait. But what, kind of takes it forward and allows us to have the true foundations of modern genetics is being able to then lay out why this must be a recessive trait and why it has to occur on the X chromosome. It has to be on the X chromosome, of course, because, well, the white eyes are eventually observed in females, so it can't be on the Y chromosome. And once you know it's on the X chromosome, you can work out the details and see that it has to be recessive, etc. So this brings us to a problem. And the problem is, well, it's trying to point out that there are conditions here, right? So, you know, a lot of your probabilities are conditional upon whether you're working with a male fruit fly or a female fruit fly. And so it's not surprising that we're going to have the probability of an event A given some other event B. And the definition of this conditional probability is, well, if I'm given B, I'm restricting myself to the collection of events B that may have a huge probability, it may have a small probability, but that's what I'm going to divide by. And I'm going to take the probability that A and B both occur and divide that by that probability. If A and B are independent events, we've seen this in a previous problem where independence means that the probability of A and B occurring is equal to the product of the two probabilities. So if you think about if the events are independent, subbing this in for probability of A and B, well, you're going to have a probability of B divided by a probability of B, and so you're just going to get that the conditional probability is the probability of A. That's nice. It's very intuitive. It's saying that if two events are independent of each other, not disjoint, but independent of each other, meaning that, the likelihood of one doesn't affect the likelihood of the other. If that's the case, then if you know that B is true, it doesn't impact the likelihood of A occurring. So that's very natural and intuitive, and it's nice that the mathematics reflects that intuition. We want to come back and say, well, how does this connect to sex-linked traits? Let's think about the concept of genetic equilibrium, where we're not changing the allele frequency over time. Everything is uniformly distributed. And genetic equilibrium would say, well, the alleles for this gene are uniformly distributed over the males and the females. So it's this distribution of alleles is independent of sex. That's what we need to specify. So if that were the case, well, what we would say is we're given an array, and it's similar to what we've seen in a previous problem, where the array has length n. A of k is the proportion of males that would exhibit the kth out of n total recessive. And we're going to say it would need to be an X-linked gene because, well, if it's a Y-linked gene, we're certainly not going to have independence here. So what's the proportion of males that have the kth of these n totals? And so we're in genetic equilibrium, meaning the likelihood that an X chromosome has this recessive allele doesn't matter whether it's male or female. But what we're going to do then is say, what's the likelihood that a randomly selected female will be a carrier for this gene? So what it means is, we can measure essentially the number of males that have the trait. That's what's nice about an X-linked gene is that in males, they only have one copy. So if it's recessive, it doesn't matter. There can't be any dominance over that recessive allele. So this is similar to what we saw in a previous problem where we looked at the number of individuals with a genetic disease. Then we estimated the number of carriers of this disease. Here, we're saying, well, if we were on the X chromosome and we see the percentage of males that display this, this is telling us the probability P of seeing that allele somewhere in the population for each of these genes. So it could be 10%, it could be 50%, it could be 80%. Some of the females will also show this, but remember, the females need 2 copies of this. So if the females need 2 copies, we're interested in not that they have 2 copies, but that they have 1 copy. So where are the females that are silent for this particular allele? So by carrier, we mean you have the recessive allele, but you don't actually have 2 copies of the recessive allele. So you have exactly 1 copy. And what this is telling us is if there's 10% of males, 50%, or 80% that have a recessive copy of this allele, then the percentage of females that will be carriers would be 18%, 50%, or 32%. So it's an interesting question, again, because, well, we get to use math, we can measure what we observe in the population, and we can infer something that is invisible to us just from what we can actually see and from an understanding of the underlying biology. Pretty cool. We just then need to write out what it is that we're computing. So in this particular case, we said, we know that the males have 1 X chromosome. So proportion of the recessive allele in the population is P, which is equal to the percent of X chromosomes. with the allele. So P is what we observe as the frequency of the recessive trait in males, because they only have one copy. And so by genetic equilibrium, P is the proportion of the recessive allele in the female X chromosomes too. So I think that's not that strong of an assumption. It's saying, females and males don't have a different proportion of recessive versus dominant alleles. I think that's probably reasonable for a huge collection of different X traits. The next thing that we might compute is to say, well, the probability that a female is not a carrier is therefore equal to two possibilities. So it could be the probability of homozygous dominant plus the probability of homozygous recessive. Okay. And so this means 1 minus P is the proportion of the dominant allele in the population. So the probability, this probability that we care about here, that you're not a carrier, is therefore equal to, well, what's the probability of it being homozygous dominant? It's the likelihood of picking two X chromosomes from the population and getting dominant alleles both times. So it would be 1 minus P squared. Similarly, the probability of being homozygous recessive would be the likelihood of picking two copies of the recessive allele. So we have probability P for each occurrence. So we have a P squared. And I'll parenthesize it in this way. We don't need to by order of operations, but just to say that this is the probability that we might be interested to saying, what's the likelihood of the female not being a carrier? What we want is the probability that the female is a carrier, meaning that they are heterozygous for this trait. So they have one but not two copies of the recessive allele. And, we could write that in one of a couple ways. I think it's just easier to say, clearly this is the complement of the previous thing that we saw. So this female is not a carrier. And so I'm just going to sub in that this probability that we care about is 1 minus the expression that we had before. And then we're good. So again, understanding the biology, appreciating the math, working out the details, making a little bit of a simplifying assumption allows us to say, what is it that we can measure, which is the frequency with which males have this X-linked trait, and what can we infer, the probability that a randomly chosen female would be a carrier, something that we can't visibly observe. We, of course, also could just simplify this. That's the other nice thing about math, is that sometimes things just will fall out from what we study. So if we wrote this out and we wanted to say, let me make sure that I'm being careful here and parenthesizing appropriately. Okay. We're taking 1 minus this expression. So we could expand out the 1 minus P squared. And then that 1 minus P squared would be 1 minus 2 times P plus P squared. And then we get a plus P squared. That's our expression here. So we're going to get a 2P squared here. We'll get a minus 2P here. And when we do the same thing, the ones are going to cancel. So these will cancel out. And we wind up with minus of a minus, which is a plus 2 times P, and a minus of a 2P squared. So 2 times P minus 2 times P squared. And that in turn can be simplified just a little more to say that this is equal to 2 times P times 1 minus P. Nice and simple. And that's probably expression that is not immediately intuitive. You may have a very high level understanding of mathematics, maybe higher than I do, and be able to say, oh, immediately, that's an obvious formula for this. That if we had an X-linked trait and we measured the proportion P of males that exhibit that trait, that we could immediately infer from that the proportion of female carriers for this X-linked trait in the entire population would just be 2 times P times 1 minus P. I don't think I possess that level of intuition. And so, the point I would like to make is this is the beauty of mathematics, to give us a simple formula for something that doesn't necessarily give us human intuition, but allows us to take something that we could not measure and give us an expression for it. It also makes our work essentially trivial here, because remember, we're going to look at k different traits, but we're just going to apply this formula k different times. So I'll say results is a list of floats. It starts off as empty. Eventually, that's what I'm going to return. And then we're going to say for each recessive allele frequency that we see in the male recessive freaks input that we have. It's just a list of floats as input that tells us for a bunch of traits, what's the proportion in the male population for this thing that we know is on the X chromosome. And if this is what I care about, well, I may even just say, I know that that's the recessive allele frequency. I'm just going to call it P, because that's what we called it before. So if we have P, I could write out what's the dominant allele frequency, etc. But really what I want is to append, 2 times P times 1 minus P. And that's the end of the story. So we can now run it and see what we get. And that's the same answer that we saw before. So you have 10% of individuals have the trait in males. That means 18% of females will be carriers. It's funny, at 50% is 50%. It's also funny that at 80% of the male population exhibiting the recessive condition, 32% of the females are carriers. You might say, that's weird, it went down. And it went down because if 80% is your proportion of recessive alleles, well, a lot of those females are going to have 2 copies of the recessive allele. So having just 1 copy actually becomes more rare. We can now go to the platform and download a data set. And we're going to move it over and call it dataset.txt. And get rid of our comments around our parsing code. And run it one more time. And success again. Let me know if you have questions. WFMD coming up next. All right. WFMD is the Wright-Fisher model of genetic drift, or weapons of funky mass destruction, I think. But we've assumed previously that at genetic equilibrium, And when you look at the allele frequencies, they're fixed. They're not able to change. You have so many individuals, it's not being selected for, etc., that you can essentially say we've got 8 billion people and the proportion is 10%. And that's not changing a lot over time. In this question, we're then going to try and loosen some of the assumptions, because if we're good biologists, then we know that this isn't necessarily true. One of the things I feel like try to teach computational biology, meaning I'm coming at the field from a computational lens and a quantitative lens. But one of the things that I'm always stunned by is when I teach young people, they'll come through primary education, secondary education, and they have a sense that, oh, we love Darwin, right? And we know that all changes that come from that, power evolution, all come from natural selection, survival of the fittest. And it's funny because most people just are never taught that that's not true at all. That in many cases, you can also have changes or evolution occurring that's not due to selection, but that's just due to random chance. And so if you look at, for example, you think about Darwin and he goes to the Galapagos Islands and he sees all these different species of things that aren't present at other places. A big part of the reason for that is organisms winding up on those islands and getting separated from other members of their species. They will change over time and differentiate from those species. Some of the time, sure, that's as the result of natural selection. But some of the time, it's not. Some of the time, it's simply due to simple random chance and changes in the allele frequency over time. We'll talk in a later problem about it, but part of the reason why this can be more prevalent on islands is that you have smaller populations. So if you have smaller populations, it's much easier for random chance to greatly disrupt the proportion of your alleles. And genetic drift is the term that often is lacking in, I think, fundamental biological education, which is simply random chance producing evolutionary changes or changes in allelic frequencies. So we'd like to have a simple mathematical model, right, of genetic drift. And there's assumptions for this classic model called the Wright-Fisher model. There's 3 main assumptions that it makes. Number 1, the generations are discrete with respect to mating. What do we mean by that? Well, we mean we have a generation, and then organisms from that generation produce the next generation, which produces the next, and so forth. So there's no blending between the generations. That seems reasonable. The second assumption is we're not randomly selecting mates, we're randomly selecting alleles. So it's as if when you form a new organism, we're drawing a ball out of a bag, where that bag contains all of the current chromosomes. You're picking one. And we're going to assume that the population size is stable over time. As I said, the smaller the population, the weirder things will get. The bigger the population, the less variance you're going to see. We're going to assume that that population size is stable over time. And what we saw when we looked at Hardy-Weinberg was, I don't really care how many people are in the population because the population is huge. So all I care about is the proportion of the alleles. And I called that P. But now if you care about the population size and you know that, well, it's not an infinite population size, meaning that the allele frequency can change over time, we're now back to a binomial process. And the reason why is the population is now finite, meaning the population of chromosomes is finite. And if you think about drawing a ball from a bag where the bag is full of chromosomes and your ball is one of those chromosomes, we're now, essentially back at a binomial random variable because, well, you could have 100 chromosomes in the bag and 10 of them possess the dominant allele or the recessive allele. Say 10% have the recessive allele. You have a 1 in 10 chance of drawing that recessive allele from the bag, or it's like a coin flip that has a 10% chance of coming up heads or tails. So this binomial random variable is going to be inherently connected to this model, and we want to quantify it. So in this particular case, you have n total diploid organisms, meaning you have 2n chromosomes in your bag of chromosomes. And in that bag, you have m copies of the dominant allele. So in the bag are 2n chromosomes. M of those have the dominant allele, and the likelihood of heads is the likelihood of drawing a chromosome out of that bag and picking one that has the dominant allele for this trait at random. So it's M divided by 2N, and that becomes our P. So previously we just looked at P. Now we say P is important only because it's the ratio of the number of dominant alleles to the total number of chromosomes. Tails would be recessive. So the recessive allele would be chosen with probability 1 minus that probability P. So our question that we're interested in is to say, I'm given positive integers n, that's the population size, m, g, and k. And what I want to produce, well, for these 4 parameters, what's the likelihood that if I have n diploid individuals, meaning 2n total chromosomes, and we start with m copies of a dominant allele, then after g generations, you're going to see at least k copies of the recessive allele. So we switch dominant and recessive, it kind of doesn't matter here. So you could say recessive allele, it's just going to mean that your P and your 1 minus P are going to be switched. So if you start with M copies of the dominant allele, after G generations, what's the likelihood that you're going to have at least K copies of your recessive allele? If we think about how to solve this question, in this particular case, it seems challenging. And there's a lot to simulate here. What is hard is simulating G generations of this process. So I'll say that is hard. Much easier would be to simulate 1 generation of the process. And that's what we're going to do. So I'm just going to keep track of everything that I need to keep track of. And I'm going to pass all of my work to a function that's going to only simulate 1 generation of the process, to go from generation 10 to generation 11, where at this point in time, here's how many organisms you had, here's what your value of P was, because we're assuming that 2n is going to be fixed over time. So you always are going to have P, but the P itself is going to change because M, the number of dominant allele copies, will change based off of random chance from one generation to another. So what we will say is, at this point in time, what's the likelihood that we have some number M of dominant alleles? And if based off of that likelihood, how would that factor into the likelihood of seeing at least K recessive alleles in the next generation? Now, what will make this hard is the fact that we will have a bunch of different likelihoods based off of a bunch of different likelihoods of M. So after 10 generations, remember, it's a random process. So if you want to say, what's the likelihood of having 100 copies of your allele, of your dominant allele, it will have some probability. But you may also have a probability of 101 dominant copies, or 200, or 3,000. And each of those values gives you a different proportion of the dominant allele that's present at that point in time, and you need to sum the contributions over all of those different possibilities, where those contributions are weighted based off of their probabilities in order to give you an answer for the next generation. So that's what we will do, and this is why we would never want to try and solve this for all G generations at once. What we want to do is to say, we know how many chromosomes are in the bag, and then we're going to use that by saying, let's start with declaring the probabilities of K copies at the start before we do any simulation. So I'll say I have a distribution. It's a list of floats, and it's a bunch of zeros. Allele total plus 1, because remember, we're going to go from 0 to 2n inclusively. So the likelihood of having K copies of the dominant allele at the start here. And everybody is 0 except for the value of M. So that's going to start off at 1.0. Why? Because I'm given that I know 100% with 100% likelihood I start with M copies of the dominant allele. What you're going to see is I actually could have started with a bunch of probabilities, and I could still solve this problem. So I could have a distribution of likelihoods that you start with different values of m, but let's just assume that we know that there are m to begin with. So the distribution is going to set its value associated with m equal to 1, since that's a ground truth. Once I have that ground truth, then I want to range over the generations and advance. So I'll say 4, I don't care about the value, but I'm going to range over all the values of g. So I'm just doing something for all of the generations that we see. And I'm going to say update the distribution to be equal to the next generation of that distribution. And we need to give it the allele total. I guess you could infer it from the distribution. So a little bit redundant here. I already have the subroutine written, but I think you get the idea. The allele total is the length of the distribution minus 1. And then we say, okay, once we've done this, this has now computed, we now know, and the current, for each generation, the probability of having exactly k copies of the dominant allele. And we only really care about the final generation here. So what we will say then, and notice that k is what you're interested in. So we want to compute the probability of at least k recessive copies after g generations. And so if we want at least k recessive copies, this is equal to the probability of at most, 2n minus k dominant copies after g generations. And the reason why is you have 2n total. If k are recessive, 2n minus k are dominant. And if we want more than k recessive copies, that means we have fewer than 2n minus k dominant copies. So the distribution here really is never going to touch storing the number of copies of recessive alleles, because everything's going to work with dominant alleles. And then when we want the value that we care about, I'm going to say, we're going to range over all the values of dominant count, and I'm going to range up to, from 0 up to some particular number, and I'm going to say max dominant is equal to allele total minus k. And so I'm going to go from 0 up to max dominant plus 1. The reason why is I want to go up to and including 2n minus k. So if I want to go up to including it, I go plus 1. I don't need to start the ranging at 0. That's done by default. And as I go, the question is, what is it that I want to do? At the end of this process, I want to say, I want to sum all of the values of whatever the distribution currently is. So add the current value of the distribution into an overall probability. So I want to say, all right, I need to set a value of probability equal to 0.0. And then I want to update that probability by saying plus equal the distribution value of the dominant count. And then I eventually want to return the probability. Of course, we're assuming that we can write our next generation function that takes the current distribution of dominant alleles and models how they might change due to random chance over the next generation. So we need to write that function too. So distribution C is the probability that the population currently holds exactly C copies of the allele. What we will do is, over that distribution of possible probabilities, we're then going to recompute the distribution in the next generation based off of these probabilities. The idea is the probabilities can change, and they're going to change based off of random chance. So we want to allow this to happen. of course, the probability expressions are just based off of a binomial distribution, so they have exact computations. That's what's nice about this, is that even though there is a random process, we can say after 10 generations, what's the likelihood of having 8 different alleles? Well, it's just repeated computations of a binomial distribution formula, so it's something that we actually can compute. So it's the beauty of probability, that you have random chance, but you can also express or bound what that likelihood actually is. So we'll range over all the current counts that have a non-zero probability. We don't care about simulating anything if it has 0 probability. That's not going to do anything for us. So what I'll do instead is to say, for the current count in the range over allele count plus 1. Okay. I think I called it, oh, sorry, allele total plus 1. And the current probability I'm interested in is prob equals the distribution of the current count. I only will simulate based off of this prob if it's non-zero. And so I'll say if prob is not equal to 0. Okay, so this is essentially saying, right, like if this is current count is 20, this is saying if it's the case that I have 20 copies of the dominant allele at this point in time, I already know what the likelihood is of that. And so if it's 1 out of 100, well, I'm going to run the simulation for this particular proportion of dominant alleles, but then I'm going to weight it by 1 over 100, so that if for the next value it has a probability of 1 out of 10, well, in the next generation, the contribution of that value to the overall simulation should be 10 times as great. Or if the next one is 1 over 1,000, it should have 1/10 the influence of what we started with. So as we go through this, I will say, the success probability, should say probability weighted. It's really the probability weight. And then the success probability here is, well, it's the current count divided by the allele total. So this is saying, this is the frequency of dominant alleles for this particular value. So if there were 100 individuals, there's 200 total alleles, and if this were 40, you have 40 dominant alleles out of 100, 200. So success prob is our weight of coming up heads. So it's a value of p in the binomial process. Now this helps us understand it. So I'll say then, we will spread this probability over the next generation accordingly. So I'll range over all the alleles possibilities. Okay. And then I will say, well, the next distribution, and I need to define it. So we're going to say next distribution is a list of floats. Space there. And it's a bunch of zeros. So we start at 0.0. And that goes to allele total plus 1. And then at the end of this, that's what we return. So the next distribution is going to receive a contribution at this particular point. So it's next count that we're going to get. Okay. And here success prob is our p. And I think actually maybe I just say that this is p. I think that makes it a little more understandable, as p is such an important value as part of this binomial process. But you really need to know kind of 3 things. You need to know the total number of trials that you care about, or n. In this case, it's 2 times the number of alleles. Okay, so we know that. We need to know the proportion of things that we're drawing from. So the likelihood of coming up heads, that's P, that we know. And then we need to know the value of like K, right? And that's what next count is. That's what we're ranging over. So here we're going to say this is equal to, or plus equal to, this current probability times, actually, this is probability weight. And we're just simply going to compute this binomial that we care about. And it's going to take the allele total as our n. It's going to take next count as our k, the number of successes we expect to see. And it's going to take p as the likelihood of coming up heads. So the probability weight is just saying, well, remember that when we compute this, its contribution to this next count value is weighted based off of how likely it was that we saw this number of dominant alleles in the population corresponding to current count in the previous generation to begin with. Probably the most important thing you could do to fully understand this question is to literally write out how these things update for a small array over like 2 or 3 generations. And then that would make everything click here, because writing this out and explaining all of these specific mathematical formulas is going to be like kind of obvious and extremely tedious. But if you just write out the example, I think it makes everything really click. But what I have here is, it's just a binomial. I have an n, I have a k, and I have a p. And so if I've got the n, the k, and the p, I'm just going to call this binomial. We know what this is. We know that we're returning a combination. And that combination is going to take 2 things. So the trials that we care about is, I'm not doing this. So this is trials. And then it's choose the number of successes, which is k. So this is n choose k. And then remember, you have your success probability. So the binomial distribution says you have a success probability raised to the power of k. And then you multiply by 1 minus that success probability raised to the power of n minus k. So the number of trials minus the number of successes. And that's, again, review of what we've already seen with the binomial distribution, which we're not covering again. So then, hopefully, this will be good. What this is doing is quite complex. It's saying, here's your value of n, m, which is the starting number of dominant alleles, g, the number of generations you're going to run, and k, which is, at the end of the day, what's the probability of having at least k recessive alleles. And so we should get 0.772 for that when we run this process we do. Again, it would be very illustrative to go through this code and appreciate exactly how all these different pieces of the code make this computation, and literally do this by hand, because you only have 2 generations here, and that's enough to illustrate exactly what's going on. I feel comfortable in our code for now, though, and so I'm going to get a data set and plug it in. Trying to. There we go. It's telling us that it's 0.138 for this collection of values. Again, the power of modeling something mathematically and getting a concrete answer from it. Pretty good. And our answer is correct. Okay. Let me know if you have questions. We'll keep going. We've still got some more population genetics challenges to address. That one's probably the trickiest one. Up next is FOUN. So I'll get set up. All right, moving on. FOUN is the founder effect and genetic drift. So at this point, we've seen a little bit with respect to population genetics. We've talked about Hardy-Weinberg equilibrium. We've made a transition towards this Wright-Fisher model. And the Wright-Fisher model is getting at the idea that, Darwin's known for natural selection, but speciation is much more likely in the context of isolation. And that's because isolated populations tend to be small. So the effects of genetic drift or the random influence or the effects of randomness on allele frequencies, and which can produce evolutionary changes or changes in species that are not necessarily selective. That happens much more frequently in isolated populations because, well, if you think about this as a bag of chromosomes that you're drawing from, that bag doesn't have as many chromosomes in it. And as a result, it means that the assumption that Hardy-Weinberg would make of infinitely many chromosomes gets stronger and stronger as the population size gets smaller. So it means it will be more devolved from reality. And this is observed in human populations too. So when you have isolated human populations, you have a much greater likelihood of, a random genetic change producing a disorder that affects a substantial percentage of the population. I would extend this to say, that's called the founder effect, that you have, genetic drift that disproportionately selects smaller populations. I would extend this to say this can extend to extinction too. And that could be extinction of the organisms. I mean, in any population, there is some probability that the population itself will die out over the next generation or over the next collection of K generations. Why? Well, it could be due to predation or something. Also could just be because you don't have any mating, so you don't actually produce more offspring. Now, the likelihood of that, admittedly, for large populations is extremely tiny. And yet what I'm getting at is the same thing can apply to recessive alleles or dominant alleles, that the frequencies can change over time, but in every single generation, the likelihood of the number of recessive alleles being zero is non-zero, right? It might be tiny, but it's non-zero. And actually, on a long enough time horizon, if you have a dominant allele and a recessive allele, regardless of what your proportions are, the results of random chance are going to cause one of those alleles to go to a frequency of zero. Now, it might be that the world ends before you ever get there, but there is some probability in every generation, and over a long enough time horizon, you would eventually go to zero for one of your dominant alleles or one of your recessive alleles, even without any selection. This leads us kind of to our problem, which is that what is the likelihood after some number of generations that one of your alleles will just go extinct? So we're going to say, imagine that you start with positive integers n and m, and you have an array that contains k integers between 0 and 2n. What does that array represent? It represents the number of recessive alleles for a bunch of different factors. So you have A has length k, so you have k total factors. And every number that you have is between 0 and 2n. So that number itself is the number of recessive alleles in the population. If that number of recessive alleles is 0, it means that the recessive alleles have died out, but we're going to start off with some values that are not. So in this particular case, we have n and m specified in our sample. And the array contains 3 integers between 0 and 2n. And the idea here is, this is the number of recessive alleles at the start for the jth factor is 0 for this gene, it's 1 for this gene, and it's 2 for this gene. So notice n is the number here, which means you have 8 chromosomes. And what this is saying is, if you have 8 total chromosomes in your population, what happens if you start off with 0 of those chromosomes being recessive, 1 has the recessive allele, or 2 has the recessive allele? What are the probabilities over time that you're going to have 0 recessive alleles left? So we're going to generate a matrix that says, each row of this matrix is one successive generation. So this is after after one generation of mating. What are the likelihoods that you will have no copies of the recessive allele left? And if we start at 0, well, in every generation, you're not going to have any recessive alleles left. So we're going to take the logarithm of the probability that you have none left. The probability at every generation of having no recessive alleles left is going to be 1. Why? Because it can't come back, right? There's no more recessive alleles in the bag to draw from when we're producing new offspring. And so as a result, these logarithms are all going to be 0, and they're not going to be changing, right? So we tried to provide a sample for this question, I think, that illustrates that idea. Once you go to 0, you stay at 0. You don't come back. You can't revive this recessive allele under this model. If you start with 1 out of your 8 chromosomes having a recessive allele, you could imagine in the next generation there's probably some chance that when you produce offspring, and that you're not going to actually result in any recessive alleles left. So the idea is the population size is going to be fixed in the next generation. You're still going to have 4 individuals, which means you're going to be drawing 8 total chromosomes. So you're thinking about, well, you have this bag of 8 chromosomes. You have a 1 out of 8 chance of drawing a recessive one with each draw. And there's some probability you just take 8 draws and you don't draw any. And if that's the case, well, you have some probability of it going to 0. It's also, there is a likelihood that if you have 2 recessive alleles, you have a 1 out of 4 chance at every draw of drawing a recessive allele on your chromosome, and maybe you just never actually draw one. So it's more likely in the case of 1, which means that the log is going to be closer to 0, but it's also possible here if you have 2 chromosomes and so forth. So I like this because what it is showing is that over time, all of these columns, every column that you like, is going to head towards 0, meaning that the likelihood is heading towards 1, because we're taking the logarithm. So the logarithm of 1 is 0. So these numbers in each column are going towards 0. And on a long enough time horizon, any column would eventually go to 0, but these are just representing our probabilities. So with the problem established, it's kind of a fun, I think, problem to try and model, because, well, we're going to be back at, thinking about this in terms of binomial distributions and so forth. But what is helpful in terms of the idea is we've already learned how to model the next generation of our kind of population based off of the current generation. So we already know how to say, if at this point in time I have this number of dominant or recessive alleles, what's the likelihood that I'm going to have all the different possibilities of dominant or recessive alleles in the next generation? We already solved this particular problem in WFMD when we introduced this Wright-Fisher model. So what we will do is we'll generate this matrix of probabilities that there are k alleles in the next generation, and that's going to be very useful, because then all I'm interested in is one value in that, which is what is the likelihood of 0 copies of the recessive allele. So this is going to be our extinction log matrix. And the first thing I want to do is, well, allele total. This thing takes n as input, the number of generations we care about. and the recessive counts, which is where we start off with. So this is our list of integers. So we're modeling this for each of a collection of different possibilities, which is the number of recessive counts of chromosomes that we start with. That was our 0, 1, 2. So this is the number of individuals, which means that the allele total is equal to 2 times this, since we're modeling them as diploid organisms. We're going to build a matrix that starts off with zeros. And so we'll say that the matrix is a list of floats and is equal to empty. And then I'm going to range over all the rows. So in this particular case, each row is a new generation that we're modeling. That's what we saw previously. And so I'll say that the row I care about is a list of floats, and it needs to be a bunch of zeros. So 0.0 times length of recessive counts. Okay. So the number of columns is equal to the length of my recessive counts. We can see that if we look back at our example. So if we have 0, 1, 2, this is if you start with 0 recessive alleles, 1 or 2, the number of columns corresponds to that. So this column corresponds to starting with 0, this corresponds to starting with 1, this corresponds to starting with 2, and then the number of rows is just the number of generations that we model. So time proceeds downward. Then we're going to matrix.append that thing. And then we're going to range over all the columns. And as we range over the columns, we're going to make the values for each factor. Those are all independent. Okay, they're not going to affect each other. So to range over the columns, I'm ranging over the recessive counts. So I'm going to say for column in range over the length of the recessive counts. Let's establish an initial distribution. Okay, so our distribution is a list of floats, and that starts off as 0.0 times the allele total plus 1. We of course know there's one value with probability 1, and it corresponds to the current value of our table. Okay. So we know what we're interested in. It's recessive counts of column. So when we access that, that tells me how many recessive alleles I start with. So because of that, I set distribution of that particular value equal to 1.0. Then what do? I advance one generation at a time, recording the log of the extinction probability after each step. So here's where I get to apply my existing subroutine that I wrote from WFMD. Now, that particular function, which we can go and look at, it's called next generation. It technically was assuming that we were working with dominant alleles and keeping track of the counts of the dominant alleles. But really, there's nothing in this function, next generation, that indicates dominant or recessive. It just says for an allele that you care about, if this is the probability of having that allele in this generation, what's the distribution of probabilities for that allele in the next generation? And there's nothing that's inherently dominant or allele about that, or recessive about that. So our probability is just the proportion of, or the number of observed out of the total. And so as a result, we're going to use that to our benefit by saying, well, that's exactly what we want. Because if I want to compute this, I'll say for the current generation in the range of the generations. Okay. At this particular case, we'll say that the distribution gets updated to be the next generation based off of the distribution that we currently have. And it takes the allele total, which we technically didn't need, but it's okay. And at this point in time, the probability that you are lost is the distribution value associated with 0. Okay, so this is likelihood that there are no more copies of the allele. And I'm not even saying dominant or recessive. There's nothing important dominant or recessive in this problem. So it could be dominant, it could be recessive, doesn't matter. What I do want to do, though, is to say, once I have established this value, set this value to its appropriate place in the matrix. And so I will do that by saying that the matrix value, the row is the generation that I'm in. Remember, as we go down the rows, we increase the generations. And the column, I already know, it's just column. I already have this particular column because that's the current factor or trait that I'm looking at. And all I'm setting it to is the common log or the base 10 logarithm of this probability lost. And the only thing there is to do is just make sure that I remember at the end, I want to return the matrix. That should solve the question. Let's see. Right, we're going to Python 3, found.py, found.py. That looks awfully similar to what we saw on the platform. Okay, so far so good. I'm going to get a data set then. I'll move it over. Right, and so our population size is 17. I'll run this, then I'll interpret it. Our population size is 17. M, our number of generations, is 4. And we're starting off with 2 copies of the recessive allele or 3 copies of the recessive allele out of 34 total chromosomes. So I just want to point out what happens, which is that you have some random chance, but the idea is once it hits 0, it can't survive anymore. So as we saw previously, if you have 2 copies of the recessive allele, well, you're going to be closer to 0 when you take the logarithm because your likelihood is closer to 1. If we think about what -1 is about 1/10. So this is more likely than 1/10 that you're going to go to 0 and have no copies of your allele remaining. This is a little bit less likely than 1/10. But in the next generation, that likelihood is going over 1/10. So as I pointed out previously, regardless of what values you give, as you go down the column over all the generations, the likelihood is increasing because the log is getting closer to 0. The likelihood is increasing that you're eliminating this particular allele entirely from the population. That is the effect of having a small population to begin with. But regardless of what the size of your population is, you will always see this increasing. You could pick any allele in the human population that you like and do the same thing. The only thing is with several billion people, you're going to have 2 times that number of people when you perform your simulation. And most alleles are at high enough frequency that the likelihood is not going to be close to 1. So you're actually going to have values here that are not close to 0 when you do that simulation, but you could. And the idea is on a long enough time scale, you're eventually going to have all your values eventually get to 0. It's just going to take a really, really long time. I'll do this. Put it into the platform. Get it right, I hope. And we got this right, and I'm just going to show kind of, I guess I can change my data set if I like. I can make the population larger, and I can make these values bigger, so you're going to have much greater likelihood of surviving. You start off with a lot more copies. But you notice that they are still heading towards 0 as you go. So if we start with, let's make it 200 individuals, and we run this for 10 generations, yes, the probabilities are going to be small, meaning the logs are going to be negative. And I could run this for a lot larger values, but it's doing a lot of work behind the scenes. So it's not surprising that this is slow. Notice we are headed towards zero, right? We're nearly if you start off with 200 organisms that are diploid organisms, meaning 400 chromosomes, you start off with 20 of those, meaning 5%, but 20 whole chromosomes are recessive, then after, 10 generations, you're nearly at a 10% chance that you've lost that allele entirely. And I don't know of a better, simpler, more concrete example of how genetic drift can lead to the extinction of an allele than that. And why the population size is so important. Because if, for example, I look at this data set and I lower this to 20, and I lower that to 2 and that back to 3, when the population size is small, right, these are heading much more quickly towards 0. Even if you set this to 20, have one value. You say, well, 40 organisms and half of them have the allele. That seems pretty stable, right? Well, yeah, but after 10 generations, there's a 1% chance that you're going to run out of one of those allele copies. Again, random chance over a long time horizon can have enormous effects on evolution. And this is something that, if you don't take anything else from this, take that from it, because I encounter students who are very, very intelligent, who have a substantial biological background, and they have absolutely zero, nil, nothing, no comprehension of this effect at all as something that happens in evolution all the time. And the reason why is because, well, number one, biological education is terrible. And number two, because it's so non-intuitive to zoom our brains out over of what happens with random chance on a very long time scale. We think of everything as, being directed by something, or there being a reason for things, and randomness can't be that important, right? And we also don't do well at thinking about big picture things that happen over maybe many, many, many generations. And random chance can cause huge, huge, huge changes. So that would be if you don't take anything else from this, please take that. Okay. Moving on, we got 2 more problems today. Let me know if you have questions. We're going to look at EBIN next. Going to do a little data set cleanup. Right. EBIN is Wright-Fisher's expected behavior. And we've introduced Wright-Fisher, this idea that, yes, you might have a substantial number of individuals in your population, but at the end of the day, this idea of genetic equilibrium over time is not a perfect assumption, because unless your population is huge, random chance can cause huge changes and fluctuations over a long time span in the proportion of alleles for whatever trait that you want to measure in your population. Now, there's variation due to chance that might cause a little bit of concern, right? Because any trait, for example, can go extinct in a population at any point in time. But what we would like to do is to say something about the expected frequency. And we've already seen in a previous problem, we've talked about expected value of a random variable. So if you have a random variable x and it takes on a finite collection of values, the expected value is just the weighting of all those values of x times their probabilities. So for a simple example, if you have 3 possible outcomes of 1, 2, and 3, and the first one has probability 1/2 and the last 2 have probability 1/4, then you would say, well, it's 1/2 times 1 plus a quarter times 2, plus a quarter times 3. Whatever that is, I'm not good at arithmetic. That's the expected value, meaning on average, if you run this many, many times, what's the average that you would see over all of those occurrences? So this problem applies expected value, but to a binomial random variable, meaning if I have a coin flip and it has probability P of coming up heads, or I'm drawing chromosomes from a bag of chromosomes, where each one that I draw has probability P of having a dominant or a recessive allele, depending on which way we're studying it. If I do that many times, what's the expected value of that binomial random variable? So if I do perform many coin flips, or I draw from the bag many times, say I do it 100 times, what's the expected number of heads? What's the expected number of draws of this recessive allele from my bag? So I'm given a positive integer n, and I've got an array of length m where all these numbers are between 0 and 1, representing probabilities. And I want an array of equal length where b of k is the expected value of the binomial distribution of n. So in this sample data set, it's 17 is your total number of trials based off of these values of p. So if I use a probability value of 0.1, so 10% of my alleles are recessive, and I have 17 draws from the bag, what's the expected number of recessive alleles that I would draw as a result? How does that change if it's 20% or 30% as the probability of coming up heads? So if it's flipping a coin with 30% likelihood of coming up heads, and I flip it 17 times, what's the expected number of heads I would get? There's two ways of coming at this. Number one is there's a very intuitive notion, which is if I flip a coin 100 times and it has a likelihood of 40% in each trial of coming up heads, what would I expect to see? It probably should be 40 heads. So as a result of that, we know what it probably should be. The math should math, and the math should give us n times p. And that's what it seems to be telling us, that here's our n, here's our p. The expected number of heads in this case, the expected number of successes, is n times the probability 0.1, which is 1.7. And in each of these cases, it's n times the respective probability of 0.2, which is 3.4, or 17 times 0.3, which is 5.1. What this is saying is our intuition is right. But I don't know, I'm a math person, and I don't think just trusting, just vibing off of intuition is necessarily the right thing to do. So we could go one of two ways. I could, annotate all of this myself or try to type it out of code, but the idea is, why is it that, if we have a definition of expected value as this weighting of all of the values of the random variable times their probabilities, why is it for a binomial random variable that would be equal to n times p? Because remember, the probabilities for a binomial random variable are a little bit complicated. They've got all these, p to the k times 1 minus p to the n minus k, and then you have an n choose k term, and then you sum that over a lot of different things. That's going to be quite involved. So this is a classic computation, and I figured, why not just show you a Stack Exchange, RIP to Stack Exchange, little math Stack Exchange for this. And what this is telling us is, the person's question is, the binomial distribution says the probability that x is exactly equal to k is this thing. It's n choose k times p to the k times 1 minus p to the n minus k. And, we've seen this to death with our work with population genetics, because it's everywhere with this foundational idea of drawing alleles or chromosomes from a collection of chromosomes in our population. So this person says the expected value I know has to be NP, but the definition for the expected value is the sum of all k from 0 to n of k times the probability. And the probability is this thing. So how on earth does this thing wind up being equal to NP? This is wonderful because it shows the power of mathematics. Sometimes mathematics is just confirming an obvious intuition. Sometimes it will give us something that's an immense surprise. In either case, it's still very relevant and important. And I want to show kind of how I would come about solving this particular question. Because if I was presented with, you know, why is it, what is this equal to? The first thing we would do as a mathematician is to write down a pattern, see if we could find something that we think it might be equal to. In this case, we are almost certain it should be equal to n times p. But then why is this the case? So top answer is good. This is a standard answer that you get in a lot of different places. There are multiple explanations for this. This is a nice algebraic explanation for it. And the person's main idea, which is excellent, is to notice, well, this thing is going to have an n factorial in it. It has a p in it, right? So maybe I could just pull it out, okay? So in this particular case, this is all your probabilities here. They just get multiplied by this extra factor of k. If there is a p in my formula, and I've got an n in the formula, I don't know, maybe I could just remove them. And so first off, this person rewrites it. I believe we can rewrite. I believe we can too, because it's true. So if you think about this being a sum from k equals 0 to n, this sum starts at k equals 0, but it's multiplying something by k. So for that term, this term when k is 0 is just going to go to 0. So that will not factor. And as a result, that is no longer relevant. So our sum goes from 1 to n of the exact same thing. We didn't change anything else. We just noticed that the first term is going to be equal to 0, so we simplified it a little bit. Now that's nice because, well, when k is equal to 1 or k is equal to anything, you're going to definitely have a factor of p in there. So a factor of p can come out of this sum. It's just a sum of a bunch of things that have a p to some power in them. So pull a p out. You can also pull an n out because this is always going to have an n factorial in the top. So I'm just going to borrow that n from each of those n factorials. So what we care about is equal to an n times a p, and now this becomes an n minus 1 factorial in the top, not an n factorial. And in the denominator, you have an n minus k and n minus 1 minus n minus k minus 1. I think I've lost it. Sorry, we have, in this particular case, it would be, well, this should be n choose k. So it's going to be n, this would be n factorial over k factorial times n minus k factorial. So far, so good. And in this case, when we write this, this really would be, you would still have an n minus 1 factorial in the top. You have a k factorial in the bottom. But you'd have an n minus 1 minus k factorial in the bottom too. So I would write this a little bit differently. And when we do that though, notice the other terms, this is still 1 minus p to the n minus k, and this is p to the k minus 1. Why? Because we pulled one of those p's out. Okay. So the right side here winds up simplifying because when you do perform k equal 1 to n of this expression, it is no different from saying, well, if I started this at 1 to n, and this is n minus 1 factorial, it's really no different from this particular expression, n minus 1 choose k minus 1. And, we're just representing this thing. And these two things stay the same. And notice, well, the k minus 1 is going to make, when this is 1, it's going to make this equal to 0. And so you just wind up doing some substitutions, and the substitutions are going to tell you n minus 1, choose k minus 1 is going to stay here. This particular thing, how I like to do it, is to substitute in and say, well, if you start k at equal to 1 and you go to n of n minus 1, it's really like saying k starts at 0 and goes to n minus 1. Of n choose k. So this is actually just an n choose k, and all this term winds up being is a sum of a bunch of probabilities that constitute the entire set of all possibilities of a binomial distribution. So k could be 0, 1, 2, 3, 4, it doesn't matter. So when you sum up, this is the probability that x is equal to k. When you sum up all of those things, it just turns out being equal to 1. They do it with this clever approach, but I think it's much easier to see that this sum is just the sum of a bunch of probabilities that all have to sum to 1. So at the end of the day, what we wind up with is NP times a sum, and that sum is just 1. So when we pulled out the NP, what was left was complex and required some algebraic manipulation, but at the end of the day, it's just equal to 1. Okay, what that means is if we're solving this particular problem, then when we look at the expected values that we care about, the only thing I need to do is something quite straightforward. So I'm just going to say that the results is a list of floats, which is equal to an empty list. We range over all of the probabilities, and I'm going to append to that probability times n, and then I want to return my results, and that's it. So when I run this, we see 1.7, 3.4, and 5.1, which is what we saw before. And we're going to be able to download a data set. We'll plug it into our folder. We call it dataset.txt. Uncomment out the parsing part of our code. And we're going to run it again. And we'll see. Got an output, and we'll put it back on the platform. And we solved the problem. Good. One problem to go. One problem to go. MEND is inferring genotype from a pedigree. This problem talks about single gene disorders. They could be encoded by dominant alleles or recessive alleles. The latter case is a lot trickier because often the affected person is going to have 2 healthy carrier parents who may not be aware that they're carriers for this condition. And if an organism, or if their offspring child, has 1 copy from each of them, then they can inherit the disease. So as a result, dominant conditions are easier to get around because if you have the allele, you have the condition, and then you know that your offspring will too. This problem is attempting to estimate the probability of disease from ancestors. So if we have a family tree, for example, and there's a famous one of this with hemophilia and royals and so forth. But we've talked already about being able to infer probabilities of events based off of what we observe. So that might be whether it's a sex-linked trait or otherwise. We've talked about if you see the probability or the proportion of individuals who possess a recessive illness, what is the frequency of carriers in the population for that illness? So that's something that we've talked about being able to compute exactly under certain assumptions. This problem extends that idea and combines it, mashes it up with something that we have seen previously, which is looking at evolutionary trees. So our problem here is going to be given a rooted binary tree. It's being drawn upside down because the idea is we're flowing to an individual who is unknown. And you have ancestors that are known maybe at certain points in time, and we're trying to infer, what are the likelihoods that if these were the genotypes of ancestors, however recent or distant they were, what are the likelihoods that I will have each of 3 possibilities at this particular position, which is homozygous dominant, heterozygous, or homozygous recessive. This tree is going to be represented in Newick format because that's what we're comfortable with representing trees as on the Rosalind platform. We can convert a string to Newick format, or a tree to the Newick format, which is a string. And this particular tree that's shown here is represented as this tree. So you'll notice there's this extra AA here at the end, capital A, lowercase a. So that's that we know the genotype of one parent. But we have to go back several generations for this example to see the genotypes of these other ancestors. These are great-great-grandparents. And we know all 8 on this particular side. So the idea then is, well, if we know all this, can we filter down the likelihoods of each particular offspring having all of the 3 possibilities for this particular trait? So we could study this over 100 traits. We don't need to. This problem is saying let's isolate it down to 1, and then we could, by independence, study this over a bunch of traits. Our idea is to recurse. So we're going to find, for everything that has 2 parents with known genotypes, we're going to figure out what the offspring's likelihoods are, and then we're going to filter that down recursively. So that's pretty cool. We're going to see probability intersect with evolutionary trees, and recursion makes a lot of sense here. The first thing that we're going to do is we're going to look at data types. And data types is going to have a special new class. And that class is called a pedigree. And the pedigree is one of 2 things. The pedigree is either a label, and that label is going to be like capital AA or lowercase a or capital A lowercase a. So that's in the case that we know the exact genotype of something, then it's a string. The other possibility is it's a tuple of genotype labels from 2 ancestors. So we will infer or be able to say I am referring to each of my 2 ancestors. So if I know the exact genotypes of the ancestors, say they're both homozygous dominant, then this would be capital A, capital A, capital A, capital A. As we go down through the tree, I'm going to represent the pedigree, meaning of all the ancestors above me, what are the ones that come from one parent side and what are the ones that come from the other parent side. So when we go back and we look at the platform, we're kind of mimicking this Newick format, because the pedigree of this particular node would just be these 2 strings. This particular node would be these 2 strings, but then we combine for this node, we would combine it into, this pedigree, which is these 2, and this pedigree, which is these 2, and we successively combine all these pedigrees until we get down to the bottom, and it's capital A, lowercase a on one side, which is what we saw here, and then it's all of this, which is the combination of, the pedigrees from this node, which is left and right, and looking up, that means we're going to need to capture all of this information at that particular node. So it's a nice, simple representation of the known ancestors' genotypes at any particular node. And if you are an ancestor, then it's just your genotype. Our solution then in this particular case for MEND needs to apply that alias that we defined as a pedigree and check based off of whether it's a string or a tuple and take different actions. So our function that we want to write is called pedigree distribution. It takes a pedigree, meaning it will get converted to Newick format, but we'll pass in the root node at the start. And then it's going to produce a list of floats corresponding to the probabilities of each of these different genotypes of the individual that I'm looking at right now. And that distribution starts with homozygous dominant, then heterozygous, then homozygous recessive for that particular trait. The first thing that we'll check then to write this function is to say, am I out of leaf? Right? And if I'm out of leaf, how would I check that? I know if the pedigree that I'm currently looking at is just a string. So if it's a string, then I'm out of leaf. If it's a tuple, then I'm not. And in this particular case, well, I want to just return whatever probability 1 associated with my genotype, which is known. So we have a little global variable defined here, which is a map that says, if I'm AA, then it's probability 1 of being AA, 0 of the other two possibilities, capital. If I'm heterozygous, it's probability of 1 of being heterozygous and 0 for the others. And if I'm homozygous recessive, it's probability of 1 of that and 0 for everything else. So I just go in and access the row corresponding to this. So this is a dictionary of strings to lists of floats, but I just go in and get the value associated with the key of whatever I am. If that's the case, then I will say we're just going to return genotype distribution of pedigree. So this will be 1.0 for the genotype and 0.0 for others. Otherwise, I want to combine the results from my parents. So I'm not at a leaf, and I'll say, let's get the two parents that I care about. I have a tuple. My pedigree is a tuple. So if I am a tuple, I'll say first parent, second parent is equal to the pedigree. So what are the two pedigrees of the two parents? And let's compute recursively the distribution over the three possibilities for each parent. So I will say the first distribution is equal to the pedigree distribution of the first parent. And we're going to do the same thing for the second parent. I'm going to call it this. And change this to second distribution. All right. So that gives me the two distributions. I've applied this recursively. And then the question is, well, so this was my base case here. I've made the inductive step. I've made these calls and I've assumed that I can compute the distributions, but then I need to combine these distributions to give my own. So at this point in time, I want to mate the distributions of the parents and return it. So I have a helper subroutine called mate that will take these two distributions and combine them and do the probabilistic computations and combine them into a single distribution based off of the parents' distributions. Now we're just going to do Mendel stuff, right? So now we just need to write this function mate that takes the parents' list of floats corresponding to its, three possibilities, and this parent's corresponding to its list of three possibilities, or its probabilities associated with each of these possibilities. Homozygous dominant, heterozygous, and homozygous recessive. And we're going to return the child's genotype distribution after looking at all the possible probabilities here. So the first thing I will do is determine the probability of the dominant allele chosen in each parent. Okay, so I can get parent 1 passes the dominant allele. Lengthy variable declaration, but it's descriptive. And, well, in this particular case, it would be, it's 100% chance if the parent is homozygous dominant. So I take whatever the probability is of that. And then I want to take half of the probability of if it's heterozygous. So I will say parent 1 of 1 divided by 2. And, I mean, you could use also a map here where you just have, I don't know, this would be AA. Maybe that would be more understandable. But as long as you just always remember that this is your order, you only have 3 things. So I think this is okay for now. And we want to do the exact same thing. I want to be careful with copying code. But this is parent 2 passes the dominant allele. So it's parent 2 of 0 plus parent 2 of 1 divided by 2. Once I have that, now we will combine them and determine the probabilities. Because what's nice about this is, well, the recessive alleles are likelihoods are the complements of these things. And so I'll say, what's the probability of AA equals something? What's the probability of this? And what's the probability of double lowercase? And then at the end of this, I want to return a list consisting of probability of homozygous dominant, probability of heterozygous, and the probability of homozygous recessive. So I'll say, well, what's the chance that I got dominant alleles from both parents? These are independent trials. And so if they're independent trials, then I want to say, well, it's parent 1 passes dominant times parent 2 passes dominant. The probability of heterozygous here is 1 minus that. Actually, let me do this one first. Homozygous recessive is easier because I can say it's 1 minus this thing. And then it is 1 minus this thing. And then if I look at heterozygous, well, I could add these two things and then subtract that sum from 1. Or I could say, well, there's two possibilities here. It's, you know, and these variables are lengthy. I kind of wish I had done P1 and P2 here. I could change that. But if I think about this as P1 and this is by P2, I can say, well, it's P1 times 1 minus P2. So there's one possibility that I pick the dominant allele from parent 1 and the recessive allele from parent 2, but there's an equally likely disjoint possibility that I pick the recessive allele from parent 1 and the dominant one from parent 2. So we might want to write it maybe this way, times P2. And then it's just a matter of plugging these in. So it'll be lengthy, but now you understand kind of what we're going for here. And I'll plug these in here. I'll plug that in there. I'll plug that in there. And then the problem should be solved. So we're going to run this. Hopefully that matches for our sample. It does indeed. So I will uncomment out my parsing code. I'm going to get a data set from the platform, download it, put it into the appropriate place. We're going to call it dataset.txt. Run it one more time, and that's going to give us these values that went to output. And we solved the problem. Great. So a lot of challenges in population genetics officially solved. If there's a higher level learning objective here, I would think, well, for one, genetic drift is important. Random chance can cause huge changes in populations, given enough time, especially if those populations are small. And we can also compute things from information that seems hidden from us. So if we have limited information genetically, if we know something about those traits, often if we know which chromosome those traits are, so they might be sex-linked or they might be not, we're going to be able to infer probabilities of things that we can't observe. So the likelihood of a randomly selected individual or the proportion of individuals in a population that are carriers for a recessive single gene condition. These are fun little questions, but I think those learning objectives are more important than the fun little questions. We have a few streams remaining. I hope I'll see you soon for those. In the meantime, please make sure to like, subscribe, drop us a comment, all that good stuff, and join Philomath to unlock a whole lot more. I'll see you hopefully on our Discord or anywhere else that we interact. For now, that's it. I look forward to seeing you for a stream sometime soon. And in the meantime, all the best.